Sure. Hello, Internet. Welcome to the live stream. We're just getting set up here. How's it looking?
It's not up yet.
No?
Usually it takes about 60-second delay.
How's it looking? Nice.
So I'll start.
Cool. Sure.
Hi guys, this is Valerie Anderson. I'm the Policy and Legislation Chair for the Pine Lily chapter of the Florida Native Plant Society. We're based in Osceola County, and tonight we have John Martin, mushroom grower, mushroom expert, leader of BioBlitz teams, and just all-around great guy, doing a presentation on exploring Florida's mushrooms. And we wanted to remind you this is our election meeting. So if you're a member, a Pine Lily chapter member, you get to vote in our elections. So check your email for your ballot. Anything else I need to add to the intro?
It is raining outside.
Right.
We've had some power issues, so if the power does go out, we'll hopefully be back in a nick of time. But just a heads up in case we lose you for a moment or two. But this is the mushroom weather, so mushroom season's upon us. So very exciting time to get you guys in the loop for mushroom identification as the season progresses for Florida. So thanks for the intro, Val.
Of course.
And yeah, looking forward to it. So thank you all for joining me today. I'm John Martin, Fungi Jon, owner of Fungi Jon Mushrooms. We grow organic mushrooms here on the farm. We're not certified yet, but we're looking into that. And we do all sorts of different varieties of mushrooms here on the farm, from lion's mane, an edible medicinal mushroom, which also grows here in Florida, which I have some here of, to some culinary gourmet edibles, such as oyster mushrooms, which I have here as well. And these are two varieties that you'll actually be able to come across in the woods in Florida. So it's good to get to know them. And we also just grow them for you to enjoy, and we do doorstep delivery and offer all sorts of different products. But I'll show you a few different things that we have to offer later. But for now, I'd like to take you on a virtual mushroom walk. So what we're going to do is basically learn about mushroom identification through different photos here in this presentation that I have of Florida mushrooms that I've found throughout the past few years. These are some of my favorite photos. I love sharing them with people, and they're great examples to discuss and just share the beauty of fungi and share all the things that Florida has to offer as far as fungi goes. So welcome to Mushroom Hour with Fungi Jon. So let's go ahead and begin. So welcome to Exploring Florida Mushrooms. So here on the screen we have a beautiful mushroom, Trichlomopsis formosa. So this is actually a type of mushroom that I found at Split Oak with Valerie Anderson, I think a couple years ago. And it was towards the back of the property. I'm not sure if you have an exact location to reference, but it's like kind of like where the Split Oak is actually, on the property. But basically this was on a down log, a decaying log. And when we see mushrooms that are growing on decaying logs, that means that they're eating the wood as the substrate. So mushrooms that eat wood and decaying organic matter, they're called saprobes. So this is one of the three main categories of mushrooms and the way that they interact with the ecology of an environment. We have saprobes, which degrade organic decaying matter. We have mycorrhizal fungi, which associate with the roots of plants and trees, where there is a symbiotic relationship where they exchange nutrients. And then we have parasitic fungi, which actually weaken or eventually kill the host tree, insect, or other fungus that it parasitizes. So those are the three major groups, and I'll show you examples of all kinds of these types of mushrooms throughout the presentation today. But this one here is an absolute beauty. We can see that it has some tufts or some fibrils on the cap. We can see that it has some beautiful gills on the underside. The underside is one of the most important things that we want to look at when we're identifying mushrooms, because that tells us basically the underside characteristics, which is one of the most important things to see. It distinguishes the major groups of fungi. So we have gills on this one. On boletes and polypores, we have pores. And then on tooth fungi, such as lion's mane, we have teeth. So those are another category within the fungi as well. So let's move on to some more here. So here I am out in the woods teaching a class. So throughout the summer, I host mushroom walks, which I'd love to have you a part of. Out there is really where the true integration happens, when we're out in the woods and we're really learning hands-on. That is how I learned. I learned mycology in a very humble way, just reading books, getting to know people, exchanging information, Facebook groups. There's so much information available on the internet out there. And throughout the years of doing this here in Florida, I've really tried to condense all that information that I've found into one easy place. So now I teach classes, and you're welcome to join those throughout the year. I have an email sign up. If you just go to my website, fungijon.com, at the bottom you can just put your email in there, and then I'll let you know as soon as we have some more in-depth classes available. Today's presentation is more of an overview, but if you really want to dive really deep, then going to one of the mushroom walks or going to one of the in-depth mushroom identification classes will really get you trained to have really proficient success with mushroom identification when you're out in the woods. So this is me here using the, oh, sorry, you can go back. Cool. So this is me here using one of the reference books that I highly recommend for Florida. This is Mushrooms of the Gulf Coast States. So this book right here is one of the best books that you can get for Florida mushroom identification. It's the most recent, it's the most up-to-date. The book in my hand is another book by the same authors. This book was not out at the time of this photo, but this book is 100% for Florida. I recommend it above all. So these books, they basically have an introduction and some brief information on mushroom identification, but the main focus of these books is the species descriptions. Some of them have keys. This book does have a few keys in it. It distinguishes the different major groups here at the beginning, and then it'll take you to some keys where it basically, in the major group sections, it will have two options, and you look for the option that your mushroom has the characteristics it's describing. Then you go to the number that it's pointing to, and then it'll take you to two more options which describe the mushroom. And eventually, you'll refine your mushroom down to the one that you have or to a group of mushrooms that you'll look at. And then in the other sections here, it has basically the full description, which you can compare your finds to, and it has the photos of the mushroom as well for comparison. And the descriptions are super important. Photos don't always convey every detail, so it's always important to have something in writing or to speak about the mushroom like I will be today. So it's not just photos that we're looking at. I'll be describing some of the features that we're looking at on these fungi and some other features that may not be visible in the photos as well. But these books are absolutely a great resource for you to have both out in the woods or at home once you find your mushrooms and you want to bring them back and identify them. So we'll talk a little bit more about that throughout class, and those are available on my website. And at the end of the presentation today, I'm going to share a coupon code for you guys to get 20% off of these just for you guys. So here is a photo of Split Oak Forest, one of my favorite places to go to. It's literally 20 minutes down the street from the farm here. I'm in St. Cloud, and this is just at the border of St. Cloud, Orlando. And a very special spot. I'm sure you guys know all about it. And full of oaks, beautiful, beautiful oaks. And oaks are one of the, in my opinion, and from what I've observed, one of the best trees to be around when you're looking for mushrooms, because they have so many mycorrhizal relationships with fungi. So again, mycorrhizal relationships, if we break that word down, mycorrhizae. So myco, of or relating to fungi, and rhizae, rhizome. So mycorrhizome, fungus, roots. So we have the fungus associating with the roots of these trees, where it either ties itself around the root or it inserts itself into the root. And then basically the roots span so far, and then the mushrooms attach to those roots and span even further. So the mushrooms actually have greater reach for nutrients than some of these tree roots would actually have without their mycorrhizal symbiont. So in that process, they're actually able to uptake more nutrients, and the tree gives them simple sugars as a thank you, and they help each other survive. So there are a lot of species like this out in the woods, and we can't cultivate them yet because they're just having these complex relationships in nature that are hard to replicate in a controlled environment. So most of the mushrooms that we do find in commercial cultivation, they're grown on sawdust. So for example here, lion's mane is a sapro. And you'll find this growing on decaying wood in forest. And what we do is we basically cook sawdust, and we cook them in these bags, and then we inoculate them with the fungus that we want to grow in a laboratory. There's competitor fungi that want to take over a rich, nutrient-dense substrate. So we have to do that in a clean room. And then basically the mushrooms fruit once they digest some of that material and they really absorb the nutrients from it, and then they can fruit once the conditions are met. And right now we're seeing a lot more rainfall in Florida, and that's pretty much the number one thing that we're looking for in Florida for mushroom hunting. We just want good rainfall patterns. We could basically hunt mushrooms year round as long as there's good rain. Summer is the main season for mushroom hunting here in Florida because of that. So you might find some lion's mane or some oyster mushrooms out in the woods, or you might find some of these other beautiful fungi, such as the mycorrhizal fungi, which are associating with the trees, which you will very rarely find available in commercial cultivation, or not at all really. You may find them wild foraged in a grocery store at some point or a specialty store, but usually out in the woods is where you'll find them. So this here is one of the first varieties of mushrooms that I want to talk to you about. So this is chicken of the woods. That is the common name. And in mycology, we really want to focus on talking about things using their Latin name for identification purposes. So common names are great for practical purposes, but when we're talking about a certain species, there can be a lot of overlap with the common names. So if we want to really know the exact species that we're talking about, then we need to use the Latin names, and sometimes it can be a little bit intimidating for people who aren't used to that, but eventually you'll get the hang of it. So the Latin name for this one is Laetiparus gilbertsonii variety pallidus. Yes, that is a mouthful. I just call it Laetiparus GVP for short. And this is Florida's most common chicken of the woods species. This is the southern chicken, as you might give it a more exact common name. So it basically has these beautiful shelves. So it's a polypore. It grows in shelving formation, and then it has small pores on the underside. And if we go to the next slide here, it's a little bit hard to see in the photo, but if you zoom into it, or if you look at the underside of some of these mushrooms that grow on trees like this, or if you use a magnifier like this to really get an up-close look, this is a loupe. It has a 10 times magnification. So you can just put this at the bottom of the underside of the mushroom, and you can observe if there's pores. So just take the mushroom and go like this. Oh wow, there's tiny holes there. I wouldn't have known otherwise. So chicken of the woods has these tiny pores on the underside, and most polypores, like this one, they're going to grow on the sides of trees. Now this one is a little bit different from some other polypores because most of them are really hard, like this reishi, Ganoderma, which we'll discuss a little more later. So this is super hard. Chicken of the woods isn't that hard. It's actually soft, it's pliable, it's edible. This, some books would consider inedible, but there are proposed medicinal benefits to it. It's been used in Chinese medicine and things like that, so people make teas. We'll discuss that a little bit more. But most polypores are really hard, but there are some exceptions, and you'll see that a lot with mushrooms. It's sometimes but not always, or most of the time, but sometimes it does this. So mushrooms can not always play by exact rules. So chicken of the woods is actually a really great edible. So the Laetiparus genus has a lot of awesome species in it that are totally edible, such as Laetiparus gilbertsonii variety pallidus, which we'll find here in Florida. And you slice it up in strips like this. It looks just like chicken fajitas. Now there is a note of mindfulness with edibility and edible mushrooms, especially with one such as this one, where with all edibles, with all new foods, you want to start slowly. So if you find something that is edible and you've identified it and you're 110% sure, beyond all doubt, that this is an edible species and you know exactly what it is, and you've cross-confirmed and done all the work that you need to do to make sure of that, and you try this, you want to start slow. You want to just eat a little bit, cook it well, eat just a tiny bit, and see how you feel. 24 hours later, if you feel fine, then move on to eating more. And that's just to make sure that you don't have any ill effects or reaction to a new food, just like you might have an allergy to a carrot per se. So there are, yes, toxic fungi out there, and there are also deadly fungi out there. But there are a lot more inedible for just undesirable taste characteristics, too hard, etc., than there are mushrooms that will actually kill you. In Florida, to date, as far as I know, there are only 3 deadly toxic mushrooms out there. 1 is very common, and 2 are not as common. And they're in the Amanita genus. And Amanita genus is actually the genus with the most deadly mushrooms in the world. So that's definitely one to learn more about. Um, but here we have the chicken fajita strips, chicken of the woods fajita strips, and we're going to cook them up in some tomato sauce. So basically, I did a little bit of a tomato sauce reduction, and I went ahead and cooked them up in a skillet, and it makes like a nice chicken fricassee kind of tomato chicken action, totally vegan. So really good stuff. If you're a vegetarian or vegan, mushrooms have a whole world of exploration of flavors and diversity. So moving on to another variety, this one is the ringless honey mushroom, Armillaria tebescens. Kind of a parasite. So it degrades trees over time weakly, and then it may kill some, but it is also an edible mushroom. But like the chicken of the woods, it needs to be cooked thoroughly, and some people do have reactions to it. So I like to start off with these two to show you that yes, there are edible mushrooms, but there are precautions that you need to take with some of these wild varieties, such as these two, which are very common edible species. So don't be discouraged by the fact that there are some reports of stomach upset and things like that with some of these species. If you know what you're doing, then and you do it, gradually and not all at once. Say, don't eat a huge bowl of honey mushrooms and chicken of the woods all at once. Then you're safe. So ringless honey mushroom. So why is it called the ringless honey mushroom as a common name? Most of the other honey mushrooms in the genus Armillaria have a ring around the stalk. So some mushrooms, they have what's called a veil or a ring on the upper half, sometimes on the lower portion of the mushroom, but more often than not, it is on the upper portion of the mushroom. So this doesn't have that versus the other honey mushrooms. And it's also called a common name honey mushroom because of the kind of golden brown honey-like color that some of them resemble. These here grow in troops, not troops, they grow in clusters. So they grow clunched together where you could literally harvest it and it would be like a little bouquet all together. And that's one of the characteristics for identification of the species. And this here grows on live trees as well as decaying trees. So it works as a parasite, and then it also works as a saprobe where it can degrade the trees over time, and then also eat it while it's dead. So it has that dual nature ecological interaction. Let me skip next. And then you might actually find deer eating these. I've seen deer eat them plenty of times, also squirrels. There's plenty of action out there from the animals eating fungi. So you might come across a few nibble marks out there in the woods. Moving on to some suburbia mushrooms real quick. So if you're ever driving through the suburbs or some grassy lawns or just walking through the park in a manicured lawn, you might see something like this from afar. So if we go to the next slide here, we can see a closer photo here. They also grow in the woods, but it's very rare to find them in the woods. On this occasion, I did find them. And this is called Chlorophyllum molybdites, also known as the vomiter, for good reason, because it makes you vomit if you eat it. So this is the most common mushroom for non-lethal mushroom poisonings. So people will ingest this because it actually resembles another mushroom that is an edible. It's a choice edible. And then it's also growing in lawns, so say your dog might eat it or a baby might eat it or something. So that is something that is possible because it's just there. It's readily available in your local environment. So something like that unfortunately can happen. But if you're trained and you know what it is and you know to be very careful with mushrooms, then you'll avoid eating this. But this one here is very easy to distinguish if you know what you're looking for versus the choice edible lookalike, Macrolepiota procera. I don't have photos of that variety today. This is the poisonous variety here. But one thing that I will tell you that distinguishes this from most other mushrooms is the fact that it has a green spore print. So a spore print is basically, spores are the genetic material of fungi. So they reproduce via spores. Mushrooms put out thousands, sometimes millions of spores per day, depending on the variety. And they're microscopic units, much simpler in structure than seeds or eggs. They're unicellular. So in theory, they're releasing thousands of spores to ensure their survival because they don't have multiple cells to nourish that spore. It's just single celled, while seeds have multiple cells that help to protect it and nourish its growth. So this species has a green colored spore print. Spore print is something very important in mushroom identification to distinguish entire genera in some cases. So a whole genus can be crossed out by a spore print or focused on via spore print. So in bees, we have a green spore print, and that's a very rare color to find in mushrooms for a spore print color. So it has like this drab olive green color spore print. If we go to the next slide here, we can see just a little bit of tinge of the spore print shining in the very front of the mushroom there towards the bottom. I wish I had a better photo of the spore print for these. If anything, we could look some up after class, after the talk. But it has kind of like an olive green spore print. And to do a spore print, basically all we have to do is take our mushroom, we would remove the stalk, and then basically, this is my ID kit here, and it comes with all sorts of stuff, which I'll get into after I show you this. But basically it includes this foil. So basically aluminum foil is not white and it's not black. So you can see white and black spore prints on it. So it's better than a piece of computer paper. Because a lot of mushrooms have a white spore print. So what you can do with the foil is take a little piece of this, just rip it off from the kit, or just bring some foil with you. And then take your mushroom, take the stalk off, put it with the gills facing down. So you're going to see the top of the mushroom if you're looking down on it. And then put a cup or just something to retain some moisture over it. And then within the next 12 hours, you should see a color on this when you remove the cup and the mushroom. And that's your spore print color. So then when you're going through your identification characteristics of the species you think you have, or if you're going through the keys, it'll say spore print this color. Then now you know the spore print. So very important thing for mushroom identification to understand how to do. And very important in the case of Chlorophyllum mollidites, the species that we have here on the slide, to make sure that it is not one that you want to eat. In here in the ID kit, we have a few different things here. So these are different reagents. So these, when applied to the surfaces of mushrooms, they turn a different color. Some of them don't have any reaction, but in the books it'll say, for example, when you apply KOH, potassium hydroxide, then it will turn olive red on the cap. So that's kind of like a further confirmation to make sure that you have the mushroom at hand. And with a certain group of mushrooms, the boletes, there's actually some that look super similar macroscopically. So either by reagents or by microscopic observation, then you can distinguish those. If you don't have access to a microscope, then reagents are probably the next best step. So I make these, I mix them up, and they're at the correct dilutions for mushroom identification, so they're included in there. And then you just use some cotton swabs to apply them. And another thing that you can do with cotton swabs out in the woods is actually just take your cotton swab and brush the underside of the mushroom, and you might even just get the spore color on that from the underside. So instead of waiting those 12 hours, it's like a little hack. You can't always do that. Sometimes it's not as mature where it's already released its spores, so in that case you'd want to harvest it and then do a spore print at home or whatnot. But that's a little cheat code. It includes this little magnifier, so it's kind of like a cheaper version of this. So you could just use this, put it up to your mushroom, and then you can see a little better the underside and see what is going on under there, so you can get your major group categorized. And then the ID kit also includes some handy dandy brown paper bags, the best storage device for your mushrooms so that they don't rot in plastic and so that they can breathe a little bit. So highly recommend bringing some brown paper bags with you. That's always in the kit in case you forget. And then it includes a full walkthrough and cheat sheet. So this is going to basically have all the characteristics of mushrooms that you need to observe and compare with the books. So for example here it has a diagram of different parts of the mushroom, it has the major groups. It's going to talk about the cap shape, the gill spacing, gill attachment, cap margin, all the things you want to look at on your mushrooms. And then the last page here is my walkthrough. So this is basically a quick class and a package and reminders for everything that you need to look at on your mushrooms that will help you identify them, both on the field and at home. And then some recommended reference books, including online resources, and just some tips and reminders, and basically how to make a spore print, just like we discussed, but a quick reminder there. So pretty much a class in a package. Also includes a little dictionary. If you ever need to reference some terms, say for example, if you're looking in the book and you don't understand the term it's talking about in a description, then you can reference this. So that's my ID kit. So if you're ever out in the woods, that's super handy. This, along with the book, super recommended. That's gonna take you above and beyond in your mushroom identification. So moving on to one of my favorite mushrooms. This is the golden chanterelle, Cantharellus genus. This is Cantharellus tenuithrix, newly named. There's many varieties of Cantharellus in Florida. And there's probably a bunch of undescribed varieties. So something that I'm doing in association with the University of Florida is actually collecting samples of different Cantharellus throughout Florida. Whenever I find them, I basically preserve them in kind of like a detergent called CTAB that preserves the genetics until it then gets sequenced. And then it gives you back a genetic code, and that goes into a database of all the varieties that have ever been documented scientifically in the world of any species. And then if we have a new species, then it won't be in the system, so then we know it's something new. So there's always opportunities to learn new things. There's so many varieties out there. It's estimated only a tenth of mushroom species have actually been documented scientifically. So there's a lot to learn, and there's a lot of ways to contribute as a citizen scientist. So definitely, do what you can to contribute when you're out there. There's iNaturalist, and a lot of people do reference that for scientific things as well. If you have, I think if you have like 3 or 4 confirmations on an observation, then it makes it research grade, and then it goes to different databases that then scientists can reference for publications and whatnot. So there's a lot of ways to contribute. But Cantharellus, awesome genus, my favorite edible in Florida. Beautiful mushroom, and it has its own kind of underside characteristic here called false gills. So like we saw on the first mushroom in the slide here, it had gills, which are basically clear-cut blades on the underside of the mushroom that the spore producing surfaces, basidia, line those gills. And then they release with moisture and maturity. With the Cantharellus genus and with chanterelles, the major group of chanterelles, we actually have what's called false gills. So instead of having those clear-cut blades, it has these forking ridges, also known as blunt flaps, or just simply false gills. So if it's a chanterelle, it will have some form of forking underside. This variety usually smells a little bit fragrant, kind of described as like an apricot-like odor. The interior flesh is white. There is a look-alike that has true gills and has an orange internal flesh, and that one is poisonous. So that's something you definitely want to look for. So with most mushrooms, I find, at least the mushrooms that we're discussing today, there's usually 3 to 5 main characteristics that if you have those all in conjunction, it's going to basically cross out pretty much any other mushroom that you would think is similar to it in some way. So in this case, we have false gills, we have apricot-like aroma, we have the bright yellow fruiting body, we have the fact that it grows around trees and not out of wood, so it's a mycorrhizal fungus. And what else am I missing here? The white flesh, I don't know if I said that already, but if I did, sorry. While the look-alike has orange flesh, orange cap, and fruiting body. It has, it grows on wood, on decaying wood, so if you dig up the base, it's going to actually be growing on like a decaying log or on some form of wood, while the chanterelle is going to be growing in the soil around the trees because it's just connecting through the mycelium to the roots. So it's more of a distant connection to the roots. And then it's going to have, the lookalike doesn't have that odor either. The lookalike actually even glows in the dark, so that's an interesting characteristic of some mushrooms. That's why it's called, Amphilotus illudens is the lookalike, called the jack-o'-lantern mushroom. And both of these varieties grow at split oak, just for example, so they grow in the same environment. So very interesting, but definitely be careful. Here's another photo of them. I love taking photos like this where it shows all different angles. We can see that the gills slightly run down the stalk on this chanterelle here. We see some kind of pitting formation on some of them, and some of them are more smooth, so that's something to just observe and document some of the differences there between the specimens. Whenever we're finding mushrooms and trying to identify them, we want to actually collect several specimens, not just one. Say, for example, if you find one that's on the older side, it may have pretty different characteristics than one that's on the younger side, and ones that are at the same maturity might even have different characteristics as well. If you were to just pick one and maybe the color got washed out or something by rain, or maybe a palm frond landed on it and some of the color from the palm frond, some tannins leached into it or something, then it alters the color. So having multiple specimens help you to just see the different variables in the traits that a certain species or the certain assumed single species is exhibiting. Assumed single species because it may actually be two separate species in some cases. There was one instance where I was finding boletes and they looked identical to the eye, but then when you use the reagents, then it's a totally different species. So you may have a totally different species. So that's something just to be aware of there. With the chanterelles, for example, as far as I know and as far as I've ever heard, there is no toxic chanterelle, but there are lookalikes such as the Amphilotus illudens that are toxic. So within the group, as long as you have the characteristics of the group and if you're looking for an edible mushroom within that genera, then you're fine. But if you're getting something else in a different genera, then that's not something you want to consume. And here's a handful of chanterelles. Very beautiful mushroom, one of my favorites, really yummy. So next slide. So basically before and after, you can basically make really nice dishes with the chanterelles. They're great sauteed. Most mushrooms, just 5 to 8 minutes on medium-high heat, with a little bit of oil, salt, pepper, some herbs if you like, is an excellent simple way to prepare them. Pasta is one of my favorite things to do with mushrooms, just incorporate them in there, add some really great texture and flavor to it. So shroom pasta all the way. And like I discussed earlier, genetic sequencing is something that is more and more prevalent nowadays, where we're discovering more species through genetic analysis and understanding the diversity and proper taxonomic placement through that DNA sequencing and genomes. So this here, again, is the CTAB that I basically take a little sample of the mushroom and put inside, and then we process that, and then it gives you back a code, and then basically it's inserted into a database where it shows you if it's a new species or not, depending on if there's data for that variety there. So just some deeper stuff. This is another species within the genus Cantharellus, another type of chanterelle. And this variety I've never found anywhere south of Gainesville. I have seen observations in other areas on iNaturalist and mushroomobserver.com, another great resource to contribute on, mushroomobserver.com, definitely check it out. And then this one here I usually find in Gainesville. So it's a really cool chanterelle, similar characteristics as the previous species, just more of a kind of a street cone orange, neon orange color to it. Quite interesting, very delicious as well. Moving on to a few other favorite species of mine to find in the woods. This is milkcap, so totally different genus from the chanterelles. This is in the genus Lactarius. This exact species is Lactarius carugus. And if you break that down, it's a great example of how Latin names are actually trying to help us remember the mushroom. So Lactarius, if we break that down, lactate, Lactarius, lactating. So all the Lactarius mushrooms exude some form of a milky fluid, hence Lactarius. And then if we break down the actual species name, carugus, then we have carugus, which is corrugated. So if we touch the cap of this mushroom, we're going to feel small corrugations or wrinkles, and that's one of the characteristics of this species. This one has kind of like a brownish colored cap and overall brownish tan colors, has a pretty thick stalk, really meaty mushroom. If you cut the gills or you lacerate pretty much any part of it, it's going to exude some milk, some of that milk-like fluid. It's not milk, but it's a latex-like substance that actually contains the mushroom spores. It releases from the gills, it releases from the cap, it releases from the stalk. So if you cut the stalk, it'll start oozing. If you cut the gills, if you just brush your finger across them and it actually penetrates the gills a little bit, then it's actually going to start oozing a little bit of that milk. So very interesting. One thing that this one also has is a fish-like odor. So to add to that, it's kind of a little bit fishy. So if you cook this up, it kind of resembles like fish. It's like a vegan fish alternative. So it's kind of like a crabby kind of taste to it. It's not overly fishy. I really enjoy it, so it's something cool to experiment with. It's actually the first edible mushroom that I ever consumed from the wild, so it has a little bit of history for me. So this is an example of the milk-like fluid that exudes from the gills here. This is a happy lactarius. So basically you just take a pine needle or your finger and you can just put your finger across it and you can watch the latex exude. And this has pretty sticky latex, so if you touch it, it's like very interesting stuff. So moving on to the next slide here, we have a handful of some edibles. So this is really cool stuff to find in Florida. So we have chanterelles, and then we have the milk caps. In the middle, we have the species we just described, Lactarius corugus, and then to the left of that, we have another species in the milk cap genus, Lactarius. It's actually recently changed to Lactifluus, the genus. So I keep referring to Lactarius. It was all in Lactarius, and then it got reclassified into 3 different genera, and now it's actually Lactifluus genus for most of these varieties that we're talking about here. So Lactifluus corugus and Lactifluus bulimus. Lactifluus bulimus variety flavus is this one here. It has like pretty much the same characteristics as the corugus. A little bit more of a fishy odor to it, not as corrugated of a cap, if at all, and has Lactarius bulimus variety flavus has more of this kind of tan white tone to it, and then Lactarius bulimus variety bulimus has more of an orange tone to it. So there's a couple varieties within that species there. But all handful of edibles there, all in good fun in a day of mushroom hunting. So here is another Lactarius here, one of my most beautiful finds in the woods. I really love finding these. I call them Smurf mushrooms for fun. So I always joke around and say this photo, basically it took me an hour to collect all these and set them up, and then it cost me 1,000 mosquito bites. So taking nice photos of cool mushrooms doesn't always come for free. Found a feather along the way, so that was pretty cool. So it just made for cool artistic flair in the photo. But this is Lactarius indigo. So a really cool one to find. Could you guess what color the latex is? If you guessed blue, you're right. It's indigo colored latex. So really cool if you eat eggs and you cook it up with some eggs, the yellow pigment in the yolk and the blue color in the latex mix to make green eggs and shrooms. So very cool one. Really love finding these. They're not as delicious as the other species in my opinion, but they're still really cool to find. Some things that we can observe on this variety are that it has a hollow stalk. So you can see some of these stalks that I've cut. You can see that there is actually a hole in them. So within that there is a hollow center. We can also see on the mushrooms here that they have zonations of color on the cap. So if you look closely, there's small little rings all along the cap that have different shades of that blue indigo tone. And then we also have pretty close gill spacing. So the gills are not too compact where it's like almost a solid surface, but they are pretty close. So gill spacing is something that we observe with mushrooms. So something that's not really done by putting a ruler up to it or a millimeter ruler, but rather just by sight and understanding the variance of gill spacing within mushrooms after seeing them. There we go. So this is one of the red russulas. So the russula genus actually has quite an abundance of mushrooms in Florida. Very, very popular genus here. Interesting thing about this whole genus, it has very brittle flesh. And it's actually in the same family as Lactarius and Lactifluus, as we just discussed. So Lactarius and Lactifluus and Russula are all in the Russulales, so the family of Russula. And they both have brittle flesh, but the Russulas have even more brittle flesh. So you can actually take one of these and toss it at a tree and it's going to explode into a million pieces. So that's kind of a fun thing to do, and it's not unmindful because you're actually helping the mushroom spread the spores, which is actually its main purpose, at least from its biological standpoint. So kind of a fun variety to work with. Not edible, as far as I know. It's very difficult to really identify these to an exact species. There's just so many. You need a microscope in order to really get down to the exact species with this red russula complex. There are other russulas out there that are a little bit easier to identify. I think we have another one coming up here. No, it doesn't look like it. Can you go back a few slides? One more? There's some easier russulas to identify, but this one is the most common. It's just red russula for common name because we don't know the exact species, and yeah, you can go ahead to the next slide. So this is a good example of gill attachment here, where it has attached gills. So whenever we see this, this would be considered attached, where the gills are actually touching the stalk. So a lot of russulas have this, touching the stalk in that way. That's one of the main characteristics that we want to look at when we're identifying mushrooms. If we look in the species description, under gills, it's going to say gills attached, gills decurrent, gills subdecurrent, gills free, which basically means that there is no attachment at all. So that's something we want to observe, and with the russulas, we're going to see attached gills. The squirrels out in the woods really love russula and lactarius, so I've actually seen several squirrels up in the trees nibbling the caps, and then I'll often find just stalks along the trail, basically the remnants of what the squirrel didn't want to eat, kind of like mushroom bones. So it's kind of interesting, but squirrels are cute, so I thought I'd throw that in there. This is an example of when mushrooms don't play by the rules. So sometimes mushrooms want to grow through concrete. So this here is Macrosby titans, one of the largest species known in North America. So it can grow really big. I've seen photos of people holding some that are literally like this big, like the size of my torso. So very big mushroom sometimes. This one was about this big, maybe a foot cap, and then maybe a foot tall, maybe a little under a foot, but a really meaty mushroom. Now I wouldn't recommend eating a mushroom that's growing out of concrete, but this is an edible variety. It's usually a little tougher. I'd assume it's even tougher growing through concrete, but an interesting one nonetheless. Macrosby titans. This is one of my favorite photos that I've taken of fungi in Florida. This is Hygroscopy cuspidata, a magical wax cap. And they're called wax caps because, as you can see, when the caps are wet, they're waxy. Very interesting variety. Has kind of these, if you look closely in the photo there, you can see some small kind of twisting fibrils along the stalk. So that's a kind of a interesting characteristic that this one has, along with the bright red cap, the bright yellow stalk, and the waxy, called viscid texture, which means it gets kind of waxy or kind of slimy when wet. This here is Amanita persicaina. Amanita identified this mushroom. So Amanita persicaina is one of my favorite mushrooms to find in Florida because it's basically the closest thing that we get to the archetypal mushroom, the Amanita muscaria. So Amanita muscaria is basically what the emoji mushroom icon is resembling. It's basically what Super Mario is chasing after. So basically, it's the mushrooms that make you power up and grow big in Super Mario. And in the case of these, some people report some effects like that where they feel very big if they consume these. So this is a psychoactive mushroom, but it's also a toxic mushroom. So if prepared improperly, then it can create toxicity. They're totally legal to possess, and that's probably because they create a little bit of an uncomfortable experience. So if you find these, I would just suggest observing them. They're a very beautiful mushroom. Some people do go and use these in the kitchen culinarily, where they will parboil them, drain the water, and then consume them. And in that parboil, it actually removes the toxins and the psychoactive compounds so that you don't have this weird experience of feeling kind of drunk and really big. And basically this one here is Florida's closest thing to Amanita muscaria, Amanita persicaina. And the Amanita muscaria would have like a bright red cap, and then this one here has more peachy colored orange to yellow tones. Something that we see here on the caps is the patches or warts. It's basically remnants of the universal veil. So that's a characteristic in the genus Amanita and in other genera, where it's basically the structure that envelops the mushroom while it's in the egg stage. It's like a thin membrane. And then as it grows, it pops through that. And with some varieties, little pieces of it stay stuck to the cap. So that's what makes that patchwork on the cap. With these, you can actually remove those pieces of warts on it. With some mushrooms, it's actually part of the cap's texture. So that's a distinguishing factor there. Warts versus scales. So if we actually feel texture on a cap and it's not coming off, then that's scales. And then here we have patches or warts, which literally come off, and they kind of look like little globules of cottage cheese on the cap. So very interesting. On the mushroom to the right in this photo, we see a ring on the lower portion of the stalk. And that is also a characteristic of the universal veil, actually the partial veil, which is the thin membrane that covers the gills while the spores are still immature. So, for example, if you ever see a cremini mushroom in the store, you can't see the gills. There's a membrane covering them. While on the portabella, you can see the gills, they're black from the spores. And these are the same exact species. The portabella and the cremini are the same species, Agaricus bispores, just at different stages of harvesting. So the cremini mushroom has that thin membrane where it's still young, where the spores haven't released, and then the more mature form, portabella, is when the gills have opened, they're much bigger, and the spores are releasing, and it's staining the gills black. So just a little background to understand the partial veil and universal veil. There's a little bit of a closer photo there on the left for seeing the partial veil covering the gills there. And then if we look down, we're going to see the bulb. So we have this bulbous base. Most amanitas have some form of a bulb. So it's very important when we're identifying mushrooms and finding mushrooms to dig lightly around the base and just observe if there's something at the base of the stalk. Some mushrooms have kind of like a cup along the bottom where there's a thin membrane around the base. So there's different forms of cups. In these books and in my ID kit on the cheat sheet, it'll have examples of that. This would be a bulbous base, and it has some fibers on the bottom there, as we can see, kind of like a texture there towards the top of the bulb, while the Amanita muscaria has rings around the bottom. So this one has more of like loose, fibrous material on the top of the base, while the Amanita muscaria, the more red variety, which you won't find in Florida, has rings around it. Very special mushroom. I actually found this right around Christmas time. And there's some interesting correlations between the Christmas story and Siberian shamanism using Amanita muscaria, so something to look into for another day. So kind of like the previous species, this is a psychoactive mushroom. Now, this is a very common mushroom in Florida. It's a tropic mushroom that grows out of cow dung. This is Psilocybe cubensis. And some really easy identification characteristics here. It has kind of a golden brown cap that fades to white at the margin. It has a ring on the upper portion of the stalk that is usually black, stained by the spores. And it bruises blue wherever it is cut or lacerated. And that bluing is actually a result of an enzymatic reaction with some of the psychoactive compounds that are within the mushroom. Now, this is totally illegal to possess, but it's something that occurs in Florida. So take pictures and observe, but don't consume, please. Beautiful mushroom, nonetheless. Grows in cow fields, grows anywhere that there's dung, and summer is its main season. And here is a close-up of one growing on dung, so we can actually see towards the base of the mushroom there. That's called basal mycelium, where you can actually see the mycelium growing at the base. And sometimes with certain varieties, just seeing the color of that is an ID characteristic. On this mushroom here, we can also see there's some fibrils on the lower portion of the stalk. So that's a kind of a texture characteristic. And then we can also see that there is a partial veil connecting from where will be the ring in the future to the edge of the cap. So when the partial veil falls, it actually can affect both the cap margin, the edge of the cap, or the ring. Sometimes it creates little fibrils along the edge of the mushroom, and sometimes it creates a ring, depending on the species. So this one here is pretty trippy looking, but it is not a trippy mushroom. So this one here bruises blue, and it looks pretty wicked, but very different than the previous species. This one does not grow on dung. This one grows on wood, and sometimes at the bases of trees, as we can see here. I believe this is a palm tree this was growing on. This is Boletellus ananus is the species name. And it's a fun mushroom, kind of a pinkish, reddish cap with these beautiful kind of hairy scales all on the cap. And if you cut it or bruise the pores, then it's going to bruise blue. And a lot of boletes have this bluing reaction, and that doesn't mean that they're psychoactive. If you look on mushroom identification forums, pretty often people mention the bluing as a psychoactive mushroom indicator, and that's not always the case. So with boletes, in this case, this is a bolete, so it's going to have these small pores on the underside instead of gills. And I'll have some photos of the pores in next slides of some other varieties in the boletes. But the pore surface on these is basically comprised of these little tubes where the spores develop inside the tubes and then they release. So the pore surface is the main characteristic of the boletes. And versus polypores, which also have pores or tubes, these typically grow more in the ground. They're more mycorrhizal tendency, while the polypores tend to grow more on trees and shelving formation. They're usually much harder. The pore surface on the boletes also peels off from the cap. If you try to separate them, they actually peel, while on the polypores, you would literally have to cut it off with a knife. So those are some characteristic differences there, even though there is that overlap where they both have pores. Here's another cool shaggy cap bolete. This is the old man of the woods, Strobilomyces genus. There's a few different varieties in Strobilomyces. I believe this is Strobilomyces Dryophilus, but it's sometimes hard to really be 100% sure of the identification of the exact species within the genus without having a microscope. But there's no known toxic varieties in this genus, and they all look pretty similar, where they have this kind of black shaggy cap, overall kind of blackish gray colors, a pore surface that turns black when you bruise it, and then the flesh actually bruises red. So like the previous species turning blue, this one instead bruises red. So that's a pretty cool thing. I don't know any other mushroom that bruises red like that. There are some amanitas that bruise kind of pinkish, but the only bolete that I know of that bruises red like this is in the Strobilomyces genus. So a cool one. And this one and the previous species are both edible boletes. But again, you always want to be 100% sure of what you're identifying and before you ever consume it. There's a photo of the redding reaction when you expose the inner flesh to oxygen. So there's an oxidation reaction, again, with certain compounds in the boletes. Go ahead to the next slide here. This is another type of bolete. This is Tylopolis tabachinus. And in the genus, the species name tabachinus is relating to the color of tobacco, tabachinus. Tobacco brown colors on this one. Go ahead and skip to the next slide there. So a couple things in this photo here that are cool to observe about boletes are, one, the pores. And if we see towards the edge of the mushroom there, we see a different color along the margin on the pores. And that is the actual pore surface color. So that kind of whitish tan color towards the edge is the actual pore surface color. And then as we move towards the stalk, that more brown color on the pores is actually the spore color staining the underside of the mushroom. So this is an example of why you would want multiple specimens whenever you're identifying something out in the wild. So seeing that to cross-compare traits between different mushrooms of the same type can make you be 100% sure that, for example, in this case, the pore surface is the color that it is, and it's not just the staining from the spores. Something else to observe on this one is the reticulation, as it's called. It's this net-like pattern on the upper portion of the stalk. And this is something that we often see on the boletes. Some boletes have it, some boletes don't, but I see it most often on boletes than in any other genus, than any other group of mushrooms. So yeah, it's just like a net-like pattern, and in this case, it's on the upper portion of the mushroom. So that's something that we would note if we're identifying this mushroom. It has reticulation on the upper portion of the stalk. And we can see in the background there we have a pine cone, so it's growing in association with the pine trees. Here we have Trametes versicolor. This is the turkey tail mushroom. This I found in Gainesville. More often than not, I actually find the look-alike to this. I'd say 1 in every 10 times that I find a look-alike, I find a turkey tail. So much more common to find its look-alike. If you go to the next slide, it's very similar in some ways because turkey tail can have different colors like this as well. But if you use a magnifier or the magnification card in my ID kit, then you can look at the underside, and the look-alike does not have pores, while the true turkey tail does have pores. So very easy way to tell them apart. You just need something to look at them really close, especially if you're not nearsighted. So we can go back a slide here. So turkey tail, Trametes versicolor is a species name. It has these zones of these concentric bands of color. They can range from colors like this, kind of grayish, goldish, sometimes to bluish or orangish. And definitely a lot of diversity in the same species itself in the colors. And then we flip it upside down, it's going to have a white pore surface, and it's actually going to have pores. Now if we go to the next slide with the Mysterium austria, it's usually not as profoundly zoned. Usually turkey tail has more distinct zoning. And Mysterium austria, the look-alike, is more paper thin. Usually the true turkey tail, Trametes versicolor, has a little bit more thickness to it. Not a whole lot, but again, the main thing you want to look at is the pore surface on these to make sure that there is a pore surface or not. If there's not a pore surface, it is the crust fungus. What is it? Crust fungus. It's not the group, but it's in another group. It's not a crust fungus, but it's in a related group of mushrooms. But anyways, it has a smooth spore-bearing surface on the underside. So the underside is basically lined with basidia. So instead of having the spores being released through the tubes or through gills, on this one it's actually just out in the open. It's totally just exposed where it produces its spores on the underside without any structures to be protected within as it develops. So that's how it reproduces. And something I didn't mention is that turkey tail, Trametes versicolor, is actually a highly prized medicinal mushroom. So it's sold in supplements, it's used highly in herbal medicine, and there are studies that are showing higher cancer survival risk in conjunction with chemotherapy. So that's a human clinical trial that's been done, and one of the more respected and recognized studies on medicinal mushrooms in the US. So turkey tail in conjunction with chemotherapy is actually showing a higher cancer survival rate. So it's actually being utilized in some cancer therapies. So very cool one, immune system benefits, and so on. So something definitely to look into. Here we have reishi, Ganoderma genus. So we have a handful of Ganodermas here. And this is Ganoderma curtisii. Ganoderma curtisii is the most common reishi that we're going to find in Florida. There is one more reishi that's very common in Florida called Ganoderma zonatum, which I'll discuss later, and that one grows just on palm trees. While this one grows in association with hardwoods, typically I find it in association with oaks more often than not. So decaying oaks you want to look for if you're looking for Ganoderma curtisii. And this one here is also a medicinal mushroom that has been used for millennia in Chinese medicine, herbal medicine, for all sorts of things like regulating blood pressure, blood sugar, immune benefits, cancer, all sorts of stuff. I'm not a doctor. I can't suggest anything for you to use for your own health. But I will say that this is used in herbal medicine, and there's a lot of potential in these mushrooms. I'll also say that a lot of scientists also give some backlash to that because there is not enough human clinical trials done with these mushrooms where we know how much to take, how often to take them, and what exact ailments that it can treat and what side effects there are. Um, that being said, I listen to all the perspectives, and I implore you to also listen to all the perspectives and make your own decision on using medicinal mushrooms in your life. Um, but there is a lot of potential that these mushrooms are showing, and a lot of the studies, even though we don't have a lot of human clinical trial studies, there are so many studies out there in vitro and in vivo that show that there is activity in these mushrooms, and it's reflected in anecdotes and in herbal medicine and in traditional use for years. There's got to be something to it. So it's only a matter of time until we really solidify that from a scientific perspective. But again, this is something that's found in your environment. So if you are looking for something to have in your medicinal herb cabinet in your local environment, this is something, one of the most highly prized mushrooms in Florida to look for. So again, Ganoderma curtisii comes in all sorts of different colors throughout its growth. At its full maturity, it's going to look like the bright maroon colored ones in this photo. And in its younger stages, it'll look more like the yellow colored ones in this photo here. But basically, it has kind of this kidney-shaped cap to it, and it always has a stalk. So Ganoderma curtisii has a stalk. If we see closely in this photo, we see these little brown dots on some of these mushrooms here. And that's actually a type of beetle, just referred to as a Ganoderma beetle, that burrows itself inside of these mushrooms throughout its growth. So something to be mindful of if you ever collect these somewhere, then you definitely want to be mindful of preserving them correctly, where you won't have a jar full of dust that's been eaten by these Ganoderma beetles over time, which is a very common thing that happens. So if you ever find these and you want to preserve them, I suggest cutting them because they will turn into a rock after you dehydrate them whole. And then if you cut them, you can actually observe if there's any insects or anything inside that you want to get rid of to preserve your harvests well. And then go ahead and dry them and preserve them that way. And I suggest keeping them a little desiccant inside of a jar, and that usually helps to remove any excess moisture there. Just a note on collections in Florida. Unfortunately, a lot of places do restrict collections to maintain just the park in general. That's the rules of a lot of these lands. So whenever you're on any park or any natural land in Florida, definitely make sure that you're in compliance with the regulations and you understand just where to collect. Somewhere that I can really recommend to collect is Ocala National Forest and private land. So Ocala National, they actually allow for foraging, and on private land with owner permission, of course, then fair game. But definitely check in anywhere that you are going to be collecting. Just make sure that you're in compliance with the regulations there. And on mushroom walks with me, I have certain properties that I've been given access to teach on, where we do have to collect specimens in order to do that. So through an educational experience, I'll be able to take you guys out in the woods and into certain places which normally may have restrictions on collections for the purpose of teaching you. So if you ever want to come out and you're not sure really where to go, then some of the mushroom walks that I host might be a good thing to do. So reishi, if you ever find a good place to collect it, it's something that you may be interested in experimenting with for your own. Here is another photo of Ganoderma curtisii, really beautiful, different color tones here. We kind of have this purplish, maroon colors and kind of orangish and yellowish. So all these colors can be shown on the cap throughout its life cycle. Here we have an older specimen, and we see the holes on it there, which is where the Ganoderma beetles are burrowing itself inside of this mushroom. So just another little reminder there. Beautiful old mushroom. So we can slice these up, and something to do with these is make a tea. It's usually the most common and easy preparation. You basically boil these down for 30 minutes to an hour, and make a tea, and that is definitely a bitter tea, but it's definitely something that has helped me when I'm sick. It makes me feel rejuvenated and just a lot better overall. So it's kind of an adaptogenic mushroom, as it's referred to in herbal medicine. So something cool to do is some tea. You can also make tinctures with it, make a decoction, a full spectrum extract, basically doing a high proof alcohol, soaking your reishi for a month in that, then taking the the mark or the spent reishi from that and making a tea from it, then combining the two at 50-50, and then you have a full spectrum extract where it has the water soluble and the alcohol soluble compounds in there. It's a full spectrum extract. So if we move on to the next slide here, this is another type of Ganoderma. So here we have both of them. So this is Ganoderma curtisii, has a stalk, nice shiny surface. And then this here is Ganoderma zonatum, doesn't have a stalk. This one grew on oak, Ganoderma curtisii. This one grew on palm, Ganoderma zonatum. So the number one thing for identifying Ganoderma zonatum is if it looks like a reishi and it grows on a palm, it's Ganoderma zonatum. The only Ganoderma that I know that grows on palm trees is Ganoderma zonatum, and it actually kills them. So it causes what's called Ganoderma butt rot. So unfortunately, it is killing some of the palms out there, but that can be seen as a positive thing too, and creating habitats for new organisms and degrading wood to create more compost in the forest to feed other trees. So it's all a balance of nature. So mushrooms are helping to both degrade, create things to degrade, and also helping the life there to continue to exist and to flourish through the mycorrhizal connections. On the slides here, we have a very fresh and clean Ganoderma zonatum, but usually what you'll find is that the Ganoderma zonatum is actually going to look like kind of dusty brown like this, and that's just when the spores have released. This one here in the photo is very young, and we can tell even more that it's very young because of the white band around the margin. So that white band, whenever you see that on polypores, that indicates that it's still growing. So that's a happy mushroom, it's still doing its thing, it's taking those nutrients in and it's pumping that mushroom out. And it hasn't gotten to full maturity yet. So I usually leave mushrooms that have the white margin. If I'm identifying stuff, I'll pick some other mushrooms along the area and I'll let the ones that are still growing, let them do their thing. Usually by the time you pick a mushroom like this, it's already released thousands, if not millions of spores. So there really is no ecological impact, no significant impact in collecting the fruiting body of a mushroom. Now, if you were destroying forest, or if you were digging up a bunch of soil near trees, then yes, you are impacting the actual organism that is associating with those trees or that is embedded in the soil around there. So as long as we're not literally just like digging into the soil in the woods and just causing a whole mess, then it's totally benign. For the most part, it's very just benign in general to collect mushrooms. It's just the fruiting body. So underneath the ground, we have mycelium, the vegetative portion of the mushroom, and then the fruiting body is just its reproductive unit where it releases spores. And by the time we collect the mushroom, usually these spores are released in mass. But in this case, if it's a very young mushroom, I'll usually leave it so it does get that opportunity to release its spores and pass on to the next generation. So here we can see a more mature Ganoderma zonatum growing on a palm. That dusty brown is the spores. With Ganoderma, it seems like the wind currents just kind of put it back on the cap, and it just kind of gets that dusty brown cap from the spores. And the underside bruises whenever you kind of touch it or lacerate it. So I did a little smiley face on this one here. To the left, we can see a photo of the interior flesh. So the interior flesh of this one is brown. Same thing on Ganoderma curtisii, is brown interior flesh. Cool. So moving on to some more polypores. So this is a very, very common polypore in Florida. Super common. I find this almost every time I go out hiking. Here's one that I have in person here. I wish you guys could all feel it in person, but it's basically really fuzzy. It's kind of like Velcro. So it's a fuzzy mushroom. And that fuzziness, you can kind of use it as a loofah, exfoliate your skin. You can also use it as a Brillo pad, wash your dishes. But there's no use for it otherwise. It's just my fungi uses. So if you want to do something with this variety of mushroom, go for it. But otherwise, it's just a cool sapro with a really toughly hairy cap, also known as tormentos. When it's really tough hairs like this, that type of characteristic is referred to as tormentos. So that's the mycological terminology for that. But the underside here has these tiny, tiny little pores where you would use this magnifying lens to check them out. Ooh, cool, nice. And then on the photo here, we can see that there is still that white margin along the cap there. So this one is still growing and very happily so. So this photo here I actually took this on a piece of wood that I collected from the forest. I brought it back home and I put it in my closet. And this is what it looks like with the lights on. And if we go to the next photo, this is what it looks like with the lights off. So bioluminescence is something that is a common thing, more common than we would have expected as we're coming to know in these recent times, that a lot of mushrooms actually emit bioluminescence. And the theory behind that is that they're actually attracting insects that would normally emit bioluminescence to attract a mate. And then the insect would crawl along the bioluminescent underside, or the parts of the mushroom that are bioluminescing, and it would collect its spores on its body as it does that and then spread it somewhere new. So basically a false flag for reproduction in order for the mushroom to reproduce. So a little trickster there. But this is Dictopanus pusillus, or Pinellus pusillus is a shorter name for that. I think that's an updated name now. Mushroom names are changing frequently, but you can also reference the older names. They're just replaced into new taxonomic placement. But not as crazy looking with the lights on, and then you turn the lights off, and you got something pretty fun there. Next slide. Getting into some more funky mushrooms. This here is a stinkhorn. This is Clathrus columnatus. So if we break that down, columnatus, columned. So it has these usually 3 columns that connect and fuse at the top and at the base. And in the middle, there is this really stinky brown goop. And that brown goop, like the previous variety, attracts insects. So it attracts flies, which land on that brown goop, which is full of spores. That brown goop would be referred to as gleba, the interior spore mass of the mushroom. And then once the fly lands on that, it gets all sticky with its spores, and then it lands on a new pile of wood chips in your garden, and then you see them growing in your wood chips. So that's how they spread. And they make themselves known very well by their really bad odor. So if you got some stinkhorns, they're gonna stink. And then next to it on the right, we have its egg stage. So when it's really young, it just looks like these little white balls in the ground. And in China, interestingly enough, they are considered a culinary delicacy. So they actually pickle them in China, and that's on my bucket list of stuff to do for the name of my mycology. So someday I will maybe do a quick live feed video of me trying a stinkhorn egg. Yeah, that's gonna be fun. Stay tuned. Here's another mushroom common in China. This is Tremella fuciformis. You can find this out in the woods here in Florida. This is the snow jelly fungus, the common name for it. And jelly fungi are pretty cool. This one is kind of semi-translucent, really cool texture to it, and it just has these lobes. It's just basically like a little lobe of these jelly structures. And very beautiful mushroom that you can use in soups for texture. In Chinese herbal medicine, it's considered a beautifying mushroom. Apparently it improves complexion and helps your skin. I don't know too much about that, but that is what I've heard from the herbal medicine perspective. As far as identification, it's kind of semi-translucent, has these lobes, it grows on decaying wood, and the texture is just jelly-like and usually kind of white to opaque, kind of translucent. You can go to the next photo. Another photo there, same location. Actually found these towards Sanford, so north of Orlando. Pretty common in the summer to find, and you can use them in soups and stews. Pretty yummy. Not a lot of flavor to it, but adds some good texture to soups. So coming towards the end here, I want to show you guys a few species that are kind of newer to Florida. So this here is Philipsia domingoniensis, and this variety here is a type of cup fungus. Cup fungi actually produce their spores on the top instead of on the underside. And this really beautiful kind of pinkish magenta colored cup fungus is more of a newer species to us here in Florida. It's something that we weren't seeing 5-6 years ago, and we're seeing it more and more often here. These were found in Gainesville. I found these with mycologist Alan Arlene Bissett and a few other people who were on a mushroom walk there in the woods, Curtis Peer and Nicholas Thomas, I believe is his name. So we found these in the middle of this preserve in Gainesville, and it was something that these very reputable mycologists had never seen before, and they were very fascinated. So that just goes to show that there is so much out there that even experts are still dying to see and still discovering on their own and learning about. So that's one of the things that really fascinates me about mycology is that there's always more to be learned. There is never going to be an end, it seems. There's always new information, new data coming, and in this case, a new variety for Florida. If we go to the next slide here, we have an example of an undescribed species that is actually super common in Orlando. So in a lot of gardens, in a lot of parks, in a lot of just wildlife areas, I've seen this variety. This is in the Marasmius genus, but to date, there is no exact species for this yet. And it's being sequenced by Arian Farid in the University of South Florida. And he is doing some sequencing work on these, and I've submitted some samples to him, and he's also gotten samples from other people throughout Florida, and other people, for example, in Australia sent him a sample of something that resembled this, and it all matched. So this seems to have far distribution. Here it's being found, in Australia it's being found. And despite it being so common, there's still this much known about it. So something probably in your backyard yet to be discovered. So that's just the potential of mycology and something that fascinates me and hopefully inspires you to find something and maybe contribute to this wonderful field. So to wrap things up, hiking is super fun and mushrooms make it just that much funner. There's so many different colors and forms and textures and shapes, and it's cool when you can even find something to bring back home and enjoy at the dinner table as well. I would love to have you all out on a mushroom walk someday, so definitely stay tuned on that, and we can explore some beautiful places and find some fungi along the way. Here's a group after one of our mushroom walks here. I actually taught this class at Mead Gardens, a beautiful garden in Orlando, and I teach in-depth mushroom identification workshops there, usually a 4-hour class including a mushroom walk. We go through basically every characteristic of mushroom identification. We have multiple case studies of different mushrooms to really examine and explore, and everyone will be walked through the process together as groups to really understand the whole process of mushroom identification and get out in the woods for that integration as well. Go to the next slide. Something that I did cover this during the class today. So this is an example of reagent testing here. So again, I just wanted to show you guys these kits here. So mushroom identification kits, if you want basically a class in a package, this is it. So if you can't make it to a class sometime this year, then this is a great way to get yourself more of the knowledge in a very handy way, along with these books. And then, you can go ahead and skip 2 slides there. Oh, and this is an example of some of the mushrooms that I grow. So if you go onto my website, fungijon.com, I actually do home delivery. So if you're not able to get out in the woods and you want some awesome fungi in your life, I'll come pay you a visit and give you some awesome shrooms. So we grow lion's mane, and here we have lion's mane, king trumpet, black pearl, blue oysters, gold oysters, and chestnut mushrooms. And these are just a few of the varieties we grow here on the farm. Super beautiful, super delicious mushrooms. A couple more photos there of some of the mushrooms that we grow. Next slide. Cool. And there's my website there, fungijon.com. And I want to give you guys a 20% off code. Thank you all for tuning in today. If you want to get an ID kit plus any one of these books, Mushrooms of the Gulf Coast States is available on the website, and Mushrooms of the Southeast is also available on the website, and my ID kits are available on the website. So if you get either both books, which are the most highly recommended books that I can suggest for you to have as a mushroom identifier, or if you get one book plus an ID kit, you get 20% off. So pretty good deal. And on our website we have dried mushrooms, we have fresh mushrooms, and some infused products like my mushroom infused granola, great hiking snack, and some other goodies as well. But thank you so much for tuning in, guys. I wish you many happy mycological pursuits out in the woods, and I hope you guys learned something good today, and hope to see you out in the woods sometime. Thanks again.
We have a question.
Sure. Yeah, we have questions. Open up the floor.
Eleanor would like to know, when is your next mushroom walk?
My next mushroom walk. So I don't have any scheduled yet, but now that we're getting these crazy torrential rain pours, I will definitely be scheduling some, hopefully to be done by the end of June. Hopefully, I'm going to host, I'm trying to see the right date for the end of June to host one. And then I try to do at least one a month during the rainy season. So at the end of June, maybe the end of July, the end of August. But if you just go to the website, or actually you could just go to http://sub.fungijon.com, or you can go on fungijon.com and scroll to the bottom, and then there's a little box that says sign up to receive email updates, and you just put your email in either one of those, and then you'll be on my email list, and I will send you direct updates whenever there is classes scheduled. With COVID, unfortunately, it made a lot of my classes get canceled, but now that things are kind of getting back into a little bit of normalcy, we're going to start seeing more of these classes coming back in. And with the mushroom walks, they'll be independently hosted, just me, and we'll go out through the woods, maybe Split Oak or another location somewhere in the radius of Orlando, within like 30 miles or so of Orlando. You can also follow me on Facebook at Fungi Jon, as well as on Instagram at Fungi Jon as well. And on there, I also post updates for classes, and it's cool to just stay in touch that way. But yeah, is there any other questions? No? Cool. Awesome. Well, much love, y'all. Thank you all for tuning in again.